currently cancer scholar at the Georgia Tech AIMBE training. Tsinghua University, which is MIT of China. She was the #1 in her class. One out of, I guess, the 150 million people were taking exam in Tsinghua. Then came to Georgia Tech and she received her PhD. After graduation, worked for industry, played a leadership role in Bell Labs, then returned as a faculty back to Georgia Tech and Emory. She's also currently Vice President for Finances. She and I, we work very closely to build the technical committee on bioinformatics, biomedical informatics, and also the founding of journals, and also founding transaction on computational biology, which I serve as the founding editor-in-chief. Founding steering committee.
I want to thank Matt and Yasmin for inviting me here. It has been great. We already been together 2004, 15 years. When I first came back to academic from industry, actually the first person who actually have interaction was Matt. He was the one started informatics within EMBS, and that was the first meeting I have ever attended. Ever since, we jointly built a community. Now the informatics is growing within the EMBS. I'm very fortunate to have a distinguished colleague and mentors like Matt and Yasmin. Thank you very much. I know it's actually late afternoon, and I will try to keep on time. I have 25 minutes also. What I've been trying to do is, I think instead of talking about a particular AI application, I actually want to use this opportunity to share with everyone what are the challenges and opportunities when we're actually talking about applying AI to medicine. This morning, Rod gave an excellent overview about what are the opportunities. This is my area for so many years, so I will dive a little bit deeper into that, but I still will keep that picture clear. So I know the slides are almost coming up. Talking about my background, one of the most important things I learned early on in my career and study is if we actually want to translate technology to practice, especially compared to where I used to work on. I used to work on multimedia computing. Now I work in biomedicine and health. The major difference is the price we pay for making mistake in applying AI and method to application is much higher. It's life and death. As a result of it, suddenly the playing field change because the cost for risk, for mistake become critical. As a result of it, my talk and my research has always been focusing on translation, focusing on how do we actually really translate these solutions to a practice. So as a result of that, I will use this opportunity to talk about these challenges, and let's actually see how we are getting there after all these years of effort. Thank you very much. Thank you. Okay, so I actually, my talk is translating AI for medicine and health. And then we're talking about individual and population health. And then ultimately we want to deliver the P health, right? Precise, predictive, personalized, participatory. And then in the middle, that's the revolution happening 20, 30 years ago. That's actually the more biomedical engineering technology are being invented. And then in recent years, the next wave is biomedical health informatics. I actually joined the Georgia Tech as a student. When I was there was no BME. And then in the late 20 years ago, Georgia Tech started biomedical engineering. Currently in US alone, there are more than 110 biomedical engineering departments in the US. And recently, the projection is from now on for the next 20 years, it's going to be a blooming in the biomedical health informatics area. Basically, it serves the role of accelerator, speed up, and serve as enabler, make the things that used not to happen, now happening, and then also as a hub. I want to actually first acknowledge my team before I dive into detail. And I want, our research not only is sponsored by federal government, by state of Georgia, but largely also is sponsored by industry as well as a medical school. I have 29 physician friends I work on over the years, and we actually really trying to translating to make a real impact. I also want to use this time to acknowledge a community. What I'm going to share with you is definitely not a one person's view. It's actually coming from the community. So one of the main reasons is we're all being driven by this particular challenge here. We all live longer as a result of it. The healthcare cost is increasing. And meanwhile, US is facing another unique challenge. That challenge is the outcome of US healthcare is 37, the lowest among all the developed countries. So this is where I came from. I'm actually currently, I got my PhD at Georgia Tech. It's actually in the southeast of US. And then 20 years ago, this is Georgia Tech. I started actually a joint department with Emory School of Medicine. Both are top university, one's engineer, one's medicine. Currently, my department in the US, even it's only 20 years old. We just celebrated the 20-year anniversary. Our undergrad and graduate program are both #2 in US, just after Johns Hopkins. And then over the years, we have this is actually for real. It's not it's the first academic department jointly formed by 2 universities. Georgia Tech is a state university, Emory is a private university. It's for real. This is our department building. My office is here. This is on the Georgia Tech side. This is our department building on the Emory School of Medicine side. So it's really, within my department, we are having a faculty on both sides. And then we, for example, myself, I'm primary at Georgia Tech, but I'm also considered adjunct professor at Emory. So I have 2 IPs as a result of it. So currently, we have a multiple areas being developed. Some are like neuroengineer, cancer technology, immunoengineering, cardiovascular, drug delivery. But we also have some hardcore engineering. That's actually is how we are developing into this. So among which, it's actually related to imaging, sensors, and then it's also the informatics, system medicine, and then robotics. Those are the hardcore engineering. So, as Rod mentioned this morning, we want a healthy born, healthy death, right? We want a healthy aging, healthy longevity. So that's exactly what I've been involved. That's what the Georgia Tech has been leading. Our program is leading. We actually have one of the first pediatric innovation technology program in the country because it's actually built upon our partnership with the Children's Hospital. So among which we have a lot of emphasis on big data and AI. In addition to that, on the healthy longevity, healthy aging, we have an institute being formed called People and Technology because ultimately that's what's important. It's not about technology, it's about using technology to save life. Right now we're talking about healthy longevity. So that's why, as Rod mentioned this morning, right, we're actually going to this new area. It's really a paradigm shift. We call it the National Institute of Health. Really, it's the National Institute of Sickness for century, right? So now we actually reverse engineer from sickness care to the health care. We also, from engineer perspective, we're actually talking about reverse engineer. It used to be reverse engineer, now we want to be proactive. So we actually forward engineer. In this process, as I mentioned that, there's a wave last 20, 30 years of have a lot of technology being created under biomedical engineering. As a result of it, we're having yottabyte of data. Yottabyte, 24 zeros. When I was giving a summer school talk, I was asking kids, how many zeros for yotta? No one raised the hand. So really, in US alone, 2030, and not long from now, another 10 decades, projected the US alone will have a yottabyte of health-related data because of what Frank and Bridget just talked about, molecular omic data. On top of that, it's also related to Emil's talk early on, talking about sensor, right? Continuous monitoring. So as a result of it, Myself had organized a meeting talking about strategic planning for the future. I was fortunate to be invited to be among a group of distinguished colleagues. We were actually talking about what are the challenges and the opportunities. There are three groups being discussed. One is AI for big data, which I'm involved. There are also control modeling and the human technology interaction, three areas. So fundamentally, when we're talking about big data, I think the director talked about this, Manalis yesterday also talked about this, 5 E's. Many people mentioned the 5 E's. But fundamentally, people get used to N has to be big, right? Talking about population health. We're also focusing on N equal 1. N equal 1 is still a big challenge. So basically, from lifestyle, molecular, current imaging diagnosis, making relationship, all those things together, and deliver to the people. That's the vision. Among which, I'm actually summarizing these 5 challenging opportunities under AI for biomedical big data. So coming from that group, that's what we come up. Number 1, data integrity challenge. That's related to data quality. Standardization, bias of data, reuse of data, provenance, because we need a better evidence for medicine. US has proven to be, in terms of medical innovation, is number 1. In terms of healthcare outcome, it's actually number 37. And part of the reason is poor utilized evidence. It's the first one to discover, but it doesn't get translated into practice. It's slower than Europe, it's slower than Asia. So that's actually one of the reasons this become one of the challenges here. And I also show you some figure next. Second one, integration. We also always dream about 1 + 1 greater than 2, right? That's our dream. That's why we collaborate and work together. However, currently, there's already a report proving that one of the barriers for medical genomics, basically introducing arming technology to clinical practice, is actually we haven't been able to prove that 1 + 1 greater than 2. In fact, it's very interesting. Physician now can make a decision whenever the omic is presented to them, the result is opposite to their training. Now they can no longer make a decision. So that's right now it's a big question mark. It requires advanced research. Number 3, this is probably everyone knows about it. Population-based learning, it used to be now currently more than 95% literature for sure has been largely based on correlation-based study. But we also know that we have 3 doctors in our audience, right? You make a decision based on inference. Knowledge is one piece, but another piece is the inference. Unfortunately, since 1950s, 60s, this idea was first being talked about. Unfortunately, right now, less than 1 finger percentage are ever reported is based on causal inference, largely because it's simply harder to model it. And the number 4 is real-time decision making. Right now, because people keep talking about the possibility of using big data, right? Google, server, deep learning. Unfortunately, when it comes down individual patient, is that N equal 1 need to be decided. How do you use the machine learning model developed based on like offline cohort to predict individual real-time learning, a big one. Last piece is actually the interpretation and the validation. And this one is related to our physician friend. If I give you a black box AI, do you trust the AI to make a decision for you? In fact, this is I was actually being invited to serve in a group to start the AI medicine for physicians. There are more than 4,000 physician members now. And I was a PhD in that group. I was actually being invited to speak in a panel. It's very interesting. Among 4 of us, I was the one firmly believe physicians will always keep their job because we trust physicians better than Dr. Watson. And the other 3 physician panelists are the ones panicking about they're going to lose their job, largely because of the hype about the AI. But in fact, these are the 5 challenges and opportunity coming of our group. Currently, NICE already released multiple couple proposals related to addressing these challenges. But on top of these 5, I want to actually list the 3 additional ones. It didn't get into that list, but it's also very critical for translating AI. The first one is number 6, need-driven, clinical need-driven workflow matters. It's actually very well known that it's also based on the literature done by AIMBE that for the half a century since computer-aided system being proposed, less than 5% system got developed are really being put on the clinical practice. And what a waste of energy, resource, and effort. And believe it or not, this is actually talking on the street that is computer scientist holding a hammer looking for nails, right? But that's not how the real world works. So that's why this need-driven become one of the thing if we want to speed up the translation and workflow matters. And number 7, AI pipeline matters. That's where I'm going to show some harsh reality, which is actually, believe it or not, algorithm just changing one parameter, you can have a complete opposite conclusion. How do you actually deal with that issue? And then number 8 is education. So I will try to put a coherent translation of work. We come out of our center. We used to run one of the national center. In the entire process, I'm going to touch upon some of these, but not every one of them, largely because that's called opportunities, called for more research. There's no solution yet. So let's actually first talk about the number 6 related, need-driven. No holding hammer to look for nails. It has to be coming from nails first. Then we design a tool to fix it. So using cancer example, current practice, patient have symptom and doing the medical imaging. However, because late diagnosis, 1/3 of cancer patient will die as a result of poor early diagnosis. In addition to that, we have this poor like pathology, biopsy. That's the final decision on you go for chemo or you go for radiation or surgery, right? Unfortunately, we have a limited diagnosis accuracy. In addition to that, the surgical risk, if you actually solid tumor, go for surgery, remove it, they're all challenges. So what are the challenges? I'm going to put you the 2 statistics here. This is actually being shared by the chief of Emory School, the pathology. The pathology diagnosis only have a 60% accuracy. Isn't that scary? There's 40% inconsistency there. In addition to that, for all those people go through the surgery, 47 of them will have cancer coming back due to residual, largely because our imaging technology has a limitation. When you actually cannot see it. I was physically taking my student in the OR system, the operating room, having a thoracic surgeon doing a lung cancer removal. We saw how he did it. After he carved out the tumor, the cancer part based on the MRI, the next thing he did, he was using his finger to touch the neighboring area. He feel that there are 2 small lumps there. He cut it, and then we're waiting in the OR until the nurse come back and bring the result. One happened to be benign. He leave that area alone. The other one happened to be malignant. He carved more. Is that true? That's how the physician are doing it? Unfortunately, the physician's hand, the sensitivity, he was telling me if it's less than 0.5 millimeter, he can no longer feel it. That's one surgery we observed. The other surgery is brain tumor removal. And the brain surgeon said, he's lucky he can touch it. I cannot touch the patient's brain when carve it out. As a result of it, among 47 cancer recurrence, 92% came back within 18 months. I actually think about it, the brain tumor, the recovery, and the lung cancer removal recovery. That's already a long ordeal. Within 18 months, it came out back. What's the point of doing the surgery? As a result of it, let's talk about how do we use NEO to actually develop the tool. As a result of it, that's how we formed our national center. We actually developed bioinformatics, imaging informatics, and health informatics to add a quantitative, objective analysis to help find that biomarker, molecular marker. For early diagnosis is only one part of it. The other part, in fact, is for tumor margin removal. So you can actually see the margin. The hand cannot see it, but we hopefully find a biomarker labeled with imaging contrast agent so we can actually see it. And in addition to that, for the imaging, reduce that 40% diagnosis inconsistency by using imaging informatics. So now, let's dive into like some more detail. AI. I want to actually touch upon these three. As I said, I cannot touch upon all 8 at one time, but I'll try my best. In the US, it's very well known, the top like 2 cause of death, cardiovascular disease and the cancer. The third one used to be COPD. Unfortunately, CDC is in Atlanta, is our neighbor. They just reported that now in the US, medical era became number 3 cause of death. It's actually very sad. As a result of it, let's take a look what are the errors in our data. The number one I want to actually really talk about is the omic data. This is actually, I have an army of undergrad looking for research project. I give them a 40 paper coming from top journal, New England Journal of Medicine, Nature Genetics, Nature Medicine, and then they're the one assess the data, right, quality. Let's take a look. They called all the vendors, and this is the Affymetric chip. Remotely look well. I'm worried about the sound. Don't be scared because my student had sound to this. Just be prepared, maybe. We have find out that the data is noisy, like scratch, splattering, aerosol. Oh, cool. And then this one is used to have a big sound. So it's a coffee mug look. Believe it or not, 90% of biomarker developed during the first 10-15 years of the Human Genome Project have never get into the clinical trial. They fail along the way. And that's actually partially related to the data quality. In fact, in the US, there's a big incident happened with a famous molecular cancer biologist in NCI. He reported this magic biomarker. It turned out to be a noise. It was just accidentally being utilized. So that's the reason why it's like that. As I said, I'm just picking up this example paper. This is real data from famous researchers. If you look at some of the worm here, it's actually not the worm. It's actually the biomarker being reported in that paper to be able to diagnose the disease. And then for my student, we did a careful study of the data. It happened to be a noise. Okay, all later being picked up as the biomarker. So that's why data is a big challenge. In addition to that, number 7, why I was saying about the AI analysis so critical. When we're actually talking about AI, machine learning algorithm, you pull out all the computational model after you spend so much money, get patient specimen, consensus to share. You finally acquire the data. And then what do? We want to use an analysis tool to be able to look at that, right? Among all the challenges people are facing, in fact, the first challenge is related to people do not know which tool to use, which algorithm to use, right? And so, as a result of the FDA is having this challenge, they have a different group submitted a different assay. That assay is no longer just as original device, right? They actually now, the assay had to combine with sulfur together for submission, for review. And then they discovered the problem that they have all diabetes diagnosis, groups have completely submitted a different one, and they have a hard time to reproduce it. So what exactly the scary part? This is the scary part. By the way, this is the 500 cancer patients. It's already, as a 10 years, this actually data came from Europe. In the US, we couldn't get that, but Europe is more open-minded to give us data. And based on the real clinical outcome, because this is a long time past data, some patients already died, and people already survival died. It's very clear. This is what are the clinical the result. Unfortunately, if you use machine learning algorithm, just changing one parameter, one algorithm, the outcome is completely opposite from the reality. By the way, that's why I said it's no longer an imagination or speculation. It's actually reality. So this is the scary part, right? When you actually rely on AI for doing the diagnosis, it's giving you the result, the opposite from the clinical truth. Do you still have the faith to trust AI? By the way, I actually know that this AI wave is considered the third wave or the fourth wave, right? Why the 1980s, the last wave of AI didn't survive? It was very silent for many years, only until recently it came back again. What's the real reason for that? I actually believe or not, the real reason is We're lacking of successful stories to show the power of AI. And now, on top of that, if I'm actually showing you, this is, by the way, is a biomarker omic project. You change one algorithm, the clinical result and the AI give you complete opposite result. Do you trust Dr. Watson to give the diagnosis? That's become one of the challenges. As I said, why in SF we come out of this. In addition to that, what are the other challenges? I'm using cartoon to illustrate. You actually have, for example, apple represent the healthy, orange represent the disease cohort. You do all the training you do, right? And then the prevalence change. The prevalence change. For example, Mediterranean recipe is very healthy, right? And cancer occurrence may be high. Coming to here is lower. And let's take a look. What's the machine learning algorithm? The constraint. Why I was saying when n go from infinity to 1, the most critical one. What are the issues? You train your algorithm based on this. You find the best decision threshold, okay? It's like a physician has to make a decision. Machine learning has to make a decision. However, prevalence change, right? How your algorithm can adapt to that. That's actually easily said, hardly, like everyone paid attention to develop a strategy to deal with that. In addition to that, this is another challenge, right? I already used the example. For example, in Atlanta, unfortunately, we have the highest prevalence of prostate cancer among the entire US. Now you're coming to the Mediterranean, very healthy, correct? So you still have the cancer, except that when we train our machine learning, for example, if I trained using the algorithm in Atlanta, okay, and the data, now we come to apply here, what happened? It's still the cancer, but the population changed, right? As a result of it, the system failed, right? So that's why, how do you make sure your algorithm is reproducible and reliable? This has become one of the top challenges. So as a translation, focus on different focus, where we're actually looking for more reproducible result, right? So I actually just using cartoon to illustrate this reality. So if you actually, I came from Georgia Tech, I always yellow jacket is our mascot. That's why I'm using yellow to represent the black and good area. And using red to represent the fire, I get burned if you're wrong, right? And so in fact, it's again, not illusion. It's a reality. This is the same clinical trial data I mentioned to you early on to show the AI method impact on the outcome. And I also now, I'm showing you the parameter, the impact. The cross are the one happen to be that the peak performed using the whatever the training data. But exactly same 500 patients, you use the remaining, you use 300 for training, 200 for the real prediction, right? By the way, this is FDA released, they didn't release the 200 early on. We didn't have it, we only have 300, right? And the result is, the moment you move to that 200, when they release it during the prediction, blind prediction. And actually, a lot of these parameters, you chose the peak performance, it happened to be sitting on the cliff. It's easily fall off. That's actually created larger, non-reproducible results. Non-reproducible results. Basically, certain biomarker trained, when you actually apply to the real patient, you cannot get that result at all. So that's why we're suggesting you need to perturb your parameter, make sure you get to that plateau region, as what I'm showing here, okay? And the analogy for that, I always say, when you actually, as a scientist, looking for truth, right? The absolute truth, the best performance, versus an engineer designing an airplane, have to survive in the turbulence, right? And this is the strategy. You want to design, engineer an airplane that can stay in the turbulence. It may not give you the best performance for your training data, but it actually reproduce when you actually have turbulence, which means your cohort changed. The apple changed from green to red. It can still stay, okay? And so that's what we did. We actually have this FDA data coming out with that. We actually show that indeed it's sensitive to parameter. As a result of it, this is actually the NIH the data set plus the FDA data set. We tried out both data sets, and we did realize that, in fact, you have to come up with a strategy to choose the most robust algorithm that actually can predict for both data sets. We actually show the evidence that data has a bias. Same kind of data you actually cannot reproduce. And so then in addition to that, we have to come back to the fundamental interpretation. That's why you go get For every marker you find out, you want to find out not only what the expression level expressed there and the statistical significance, but also you want to do the in silico, combine and go back to the pathway to see whether they're related to cancer or not, and to see in chromosome, right, where they are, and then in the protein where they are. So that's called the in silico validation. That's the step we want to do. Ultimately, after you do that, what else do you want to do? You want to go to the diagnostic assay. You go to the imaging, right? You go ahead and synthesize that. Let's take a look. What are the challenges? So, I actually mentioned earlier on, 40% pathology diagnosis inconsistency. Why that's happening? World Health Organization defined every cancer has subtype linking to the outcome. Some have longer survival, some have short survival. And then that's why the chief came to my office. This is the data he brought to me. He was telling me that this is the cancer has a different subtype and different stages. And he was actually asking me, for me to decide whether are they the same subtype, are they the same staging. I have no clue, right? As a PhD, I tried my best. I said that this one looks to me like a different subtype and different stages. He actually said I'm totally wrong. It's actually exactly the same subtype, same stage. Unfortunately, that's the reason why 5 pathologists, 2 will make a different diagnosis because the challenge we're facing. Because you have a whole slide, biopsy slide. When they zoom in, the field of view is very small. They're looking for the area. If they happen to look at 5 different areas, right? That's the result. It's a harsh reality. So, and then what's the consequence it has for the final diagnosis? I was actually explaining this. So this is a data set that the chief gave to us. I did a training for the day 1, the same day, a batch of data, and the other day, a batch of data. He was telling me when you have a different batch of data, because they order different batch of glass slides, and that's actually, and also by different pathology, it has differences, right? We actually show that. If it's the same day, your prediction can get 80%. Unfortunately, you use one day, for example, day 2 to train, predict day 1. We say it's like a monkey can do it, it's a random choice, right? Among 50%. Unfortunately, if you do the other way around, it's actually AI does more damage than not doing it. That's what I showed you earlier on. For that omic study, now I show you on imaging. Both AI has these potential challenges. It actually can give you the wrong opposite, like outcome. So we need to actually do the data, like informatics. So that's how we do, right? The chip is very like 28,000 times like almost 15,000 a pixel. What pathology used to do, having a microscope looking at a 512 by 512, that's already huge. But really, it's only one small tile of it. We want to use the computation in imaging informatics to create an objective, labeling. We have to deal with the curse of dimensionality issues, same as omic, where the feature is much larger than the number of patient samples available. And then, however, during the entire process, it's related to one of the biggest challenges we're facing. I think a lot of people are talking about, we're actually doing knowledge, right? And modeling. Physician medicine is knowledge intensive. Unfortunately, translate the knowledge back to mathematics, the computer rule. It turned out to be one of the most difficult challenges. So this is how we work out with the pathology department. We develop a web-based interactive tool. The pathology will label the different area. We actually can use computing. Actually take half an hour, populate that labeling they did over the entire slides. Unfortunately, to be able to translate that knowledge into description is a reverse engineering process. It's very hard. I have a simple example. For the kidney cancer, there's one type called papillary. It's completely different from clear cell oncocytoma. And then the pathologist said we're looking for finger-like feature. It took my student a year, developed so many different tools, right? Computer vision, describing the fingers, right? Corners, never worked. One day, I got inspired by fractal analysis, right? The fractal. We used the fractal. Overnight, we solved the problem. So this reverse engineer translate physician's knowledge, right, their art description in mind, into mathematics turned out to be one of the most difficult challenges. And so as a result of it, we can actually, if we can describe it, then we can actually reduce the subjectivity, make it more objective. So this is some of the results coming out from our center, being used by the pathology group. And unfortunately, now we're talking about the number 5, the challenge. Interpretable like AI. Among the 2,600 features, unfortunately, only a few features pathologists are willing to validate for us. That's all related to color, morphology, related to how they do their practice. Anything beyond that, they are not willing to validate. That's actually presented one of the big challenges. So as a result of it, now we'll start using deep learning, right? Because right now we have more data accumulated and more server. We'll start using deep learning. Deep learning is even worse. The few features using shallow learner, you still can at least describe the edges. Now deep learning features, you don't even know what they are anymore. As a result of it, this is number 5, and ISF is talking about. How do we create interpretable AI? We actually developed the AI algorithm, but reversely presented it to the physician, so they can actually interpret it. So right now there are two group of methods. One is like a gradient weighted class activation mapping. This is an example to show how we can do that. And then the other one is analysis by synthesis. So if you look at what happened, so basically this is original real data. After you do all the machine learning, I'm using a synthesis to synthesize it, to deliver that feature. I know the feature, physician see the representation of the feature. Hopefully that can help them get more confidence. I already showed you the mind-boggling like a failure result of using AI. So now there's a big push for interpretable AI. And that's one of the call for proposal for NSF in pushing for this area. Now the home run, is that enough? Of course it's not enough. The real home run I want to show you, this is also the result coming from our center. After we did that machine learning, after we deal with robustness, we got a magic to biomarker, 100% mathematically. Did in silico, remember I was showing you, you link to chromosome, cancer pathway, all validated. Then we did RT-PCR, 100%. Everyone's so happy. And then physician took back, recruited 64 new patients, started applying it. This is diagnosis assay. Very sadly, almost 10% failed, 6 out of 64. What does that tell us? That tells us that validation cannot be skipped. AI-based method, despite how robust we are, how smart we are, even we did the RT-PCR, it's not enough. There's so much patient variability. That's why it's caused this challenge. So that's why home run is always critical. And then how do we deal with that? Now we're coming back to the integration. We're hoping by whole slice imaging, we actually reduce that 10% by having omic and clinical factor to be jointly added together. But however, because our patient data is limited, at this moment, as I already shared with you, there is still a big question mark on, is 1 + 1 greater than 2 or less than 2? So that's why it requires further research. So that's why what NIH is doing is excellent. They're the ones sampling all the data set, the cancer genome atlas. Now you suddenly have more availability to the data, so you can actually further research this. And so then you can use deep learning to do that work. So that's why, let me actually show you one slide, relate back to what Dr. Petit was talking about this morning, precision medicine. This is what cancer genome atlas, NCI is doing. Grade 3, ovarian cancer. That's all they can get. We can use it now, immediately, objectively label that. We have, on the left side, 3% lymphocyte, 15% necrosis, 0% stroma, 85% tumor. On the right side, 60% lymphocyte, 0% necrosis, 5% stroma, and 95% tumor. We actually have been able to make the baby step towards what's so-called precision medicine. And there are 2 steps involved going forward. Number 1, we're going to actually populate through the TCGA, right? Translate all the qualitative grading from pathologists into more quantitative, right? The grading. But now the issue is how many of those grading done qualitatively are so much different from the quantitative way, so that we can come back, give feedback to pathologists, training the pathologists to be more accurate in the future diagnosis. So that's actually where we're getting towards the P medicine area. Now, what I didn't cover 3 things, but they're all still very important. I just quickly mention this for you, so that we bring the whole picture. The first one is the causality and the versus correlation. There's this famous study done looking into the hormone replacement therapy. They find out that somehow it's actually have a lower than average coronary heart disease, woman taking that. That's the initial conclusion based on correlation, population-based data, correlation study. Unfortunately, what's the real reason? What's the cause for this? After studying, that conclusion is wrong. The correlation is wrong. In fact, the real reason is the social-economic status. It happen to be, the women are taking those, happen to be in better social-economic status. So that's the reason why it brings a major issue. Why causal inference, so critically used by physicians, are not being used? This is actually the scientific challenge, right? We have a hard time right now to have enough knowledge being curated so that we can actually use as the base for building that. In addition to that, the graph itself, as some of you us working on this area know that, it's very hard. It's an NP-complete problem. It's very hard to do that. By the way, this is not, again, an illusion. It's a reality. This is a real radiology study, imaging-based diagnosis. Actually, based on the correlation study, it's actually turned out that all the outlier was originally being said as the marker, but the reality is not. So that's why we have to look for the causality, for the inference, for the cause, beyond the correlation. So that's the one I didn't cover. Second one is real-time analysis. So this is coming from our intensive care unit. They find out that after 48 hours, this is standard protocol being inserted already for 50 years. If you get to a certain level, I actually told the story in the summer school about myself. I one time had a high fever going to the hospital, the emergency room. They drew the 18 tubes of blood. I was okay walking, and I actually passed out after 18 tubes of blood. I complained about that, but that's actually the clinical protocol. Every hospital is doing it in the US to avoid the surprises. Unfortunately, when the 48 hours later, 12 tests coming back, some patients already died. So that's why there's a need for personalized care based on the real-time N=1 diagnosis. Reprioritize 12 tests based on different patients. What are the top 3 most critical tests? So that's actually related to the NSF research area. In addition to that, one is the education. Remember I was saying, I think also Dr. Petit mentioned this morning, right? Technology needs to help real people, patient. So as a result of it, we're facing the same challenge with AI in place. One is the patient education, one is the training, the new generation of MD and data scientists. So those are the 8 challenges we're facing by talking about translating AI for medicine and health. But right now, we're still very hopeful. We hope the 4th wave of AI stay, but we need to deliver a successful story. So with that, I'm hoping to call many of you who are interested in this area to join us for this exploration. That's the reason why I'm always showing this last page. It definitely takes a village. This area is growing. Dr. Dmitry Potyazin, he left already. He was the chief for Journal of Biomedical Informatics. I was recruited by Metin and other senior colleagues to help build the community within EMBS. But besides EMBS, this is the society I'm talking about, the physicians. They have more than 4,000 physicians now looking into AI for medicine, focusing on translation. And computer science also want to revolutionize itself, right? Create a new way of looking. So this society is all the meetings. This side is all the journals. And now NSF bring in the wearable sensor. I think Emil was talking about wearable sensor, how important it is. And then in addition to that, even science community, the Golden Research Conference started this. All of this, less than 10 years old. Majority of them around 5 years old. So that's why we need a community to get educated, so we can actually tackle the challenges as I share with you, and together, hopefully, we can have more successful story on this way. Thank you very much.